Very good morning, students. Today we are discussing regarding the practical class. Okay. So in the next class, on Friday 16th, okay, there will be a first biochemistry class. Will be there. Doing that, have to present seminar. Okay. In the morning, I have to message in the group. So just go through that, and we have to prepare for all the 5 topics. Okay. So through the Google Classroom link, you have to log in and where you have to present the seminar on 16th. Okay, on first half. So there will be 2 groups, group 1 and group 2. So first half an hour, group 1 has to present the seminar, and the second half an hour, around last second half, 75 to 150 members, the each room numbers has to present the seminars on second half. Okay, second half an hour. Okay, so the other instructions will be given before the session. Okay, so today we will discuss regarding the practical class. Okay, today we will discuss regarding estimation of SGOD and SGPT. Okay, so one is by the kit method we will discuss, and one more method is a chemical method. Okay, so chemical method is by the REITMAN and FRANKEL method. So IFCC method, kit method is by the IFCC method, that is modified IFCC is nothing but International Federation of Clinical Chemistry. That is a kit method we are using for the estimations of SGOD, SGPT. So usually this SGPT and SGOD, this test will be done under the liver function test. Okay, this test will be done in the clinical biochemistry laboratory by using the kit method. But when it comes to the UG laboratories, we are following with the chemical method. Okay. So we know that, so we have various organ function tests like liver function test, kidney function test to diagnose the various disorder. So when it comes to the liver function test, based upon the various function, based upon like excretory function, based on the productions of some enzymes, so based upon some criteria it is classified into different functions. Okay, so under the liver function test we have so many tests like bilirubin, total bilirubin, indirect bilirubin, and albumin, total protein, globulins, SGOT, SGPT, alanine transaminase, GGT. These all tests will come under the liver function test. Today we are discussing aminotransferases. This other under aminotransferases, there are 2 tests will become, that is AST and ALT. AST is also called as SGOT and is abbreviated as aspirated transamine transferase and serum glutamate oxaloacetate transaminase. So this enzyme will become under aminotransferase. So one more is ALT. ALT is also called as serum glutamate pyramidase transaminase. Okay, so we know that, so like several liver cells. Okay, liver cells contains several enzymes, like both these enzymes are mainly present in the liver. Okay, so during the liver damage, these enzymes will be released into the blood. Okay, and levels of these enzymes will be increased in the blood. Okay, all these different enzymes have been used in the diagnosis of liver disorders, but commonly routinely employed these laboratory tests are majorly AST, ALT and ALT. Okay, this will be, if there is any damage in the liver, so these enzymes will be released into the blood. Okay, so during that, during diagnosis of liver disorders, usually we'll go employ for the some tests like majorly like AST, ALT and ALP. These are the 3 major enzymes tests will done under the for the diagnosis of the liver disorders. But apart from these enzymes, even we have other enzymes test will be done for diagnosis of the liver disorders, like maybe serum nucleotidase, lactate dehydrogenase, isoseparate dehydrogenase, gamma glutamate transferase. Even these four tests will be, these four enzymes will be done for the diagnosis of the liver disorders. Mainly will go employed for one, that is AST, ALT and ALP. Apart from that, even will go with the serum 5' nucleotidase, lactate dehydrogenase, isoseparate dehydrogenase and gamma, that is gamma glutamate transferase. This test also be done under for the diagnosis of the disorder. So what is the function of this? So example, like when it comes to the these enzymes will be involved in the transamination reaction. So examples for the transamination is, so this alanine will be this, as we know that transamination is nothing but transfer of an amino group from one amino acid to the other amino acid. It is nothing but a transamination. So this will be involved in transamination reaction. In presence of an enzyme called serum glutamate pyruvic transaminase, the amino group will be transferred from alanine to the pyruvic acid. So that will be forms an alpha ketoglutarate and the glutamate is an end products in presence of an pyridoxal phosphate molecule. So when it comes to one more enzyme, that is serum glutamate oxaloacetate transaminase. So this enzyme helps in transfer of amino group from an aspartate to oxaloacetate, that will be forming the end product as a alpha ketoglutarate and the glutamate in presence of the pyridoxal phosphate. Okay, this is the reactions which will be. So these two enzymes, both AST and ALT enzymes, are involved in the transamination reaction. So when it comes to the liver enzymes, though majorly I said there are three major liver enzymes. That is AST, ALT and AST. These are the three major, that is AST and ALT are the two major enzymes which will be come under the liver enzymes. Okay, based upon the liver enzymes, so for the to identification of the hepatocyte damage, we'll go for the transaminases enzyme. So we will we will go for the test transaminations enzymes test for to diagnose the hepatocyte damage. When it comes to the cholestasis, so we will go for two enzyme tests, that is for the alkaline phosphatase and the GGT. Okay, so based upon the enzymes, based upon the liver enzymes, we can diagnose the specific disorders. So when it comes to the transaminases, under the transaminases we will be having two enzymes, that is aspartate transaminase and alanine transaminase. So these two enzymes will be helping us for the diagnosis of the hepatocyte damage. When it comes to the alkaline phosphatase and gamma glutamate transferase, these two enzymes are helps in the identification of the cholestasis. Okay, these are the major enzymes which will become under the liver enzymes. So when it comes to the criteria, features of the individual enzymes, that is majorly, that is serum glutamate oxaloacetate transaminase, that is SGOT. So the normal reference range of this enzyme will be 0 to 45 units per liter. This is the normal reference range of SGOT. So when it comes to the distribution, it majorly distributed in heart, liver, skeletal muscle. So this is some distribution of the enzyme, but major activity will be this SGOT activity very high in the heart, liver and the skeletal muscle. When it comes to the lower activity, we can see in the pancreas, kidney and the skin. This is the distribution of the enzyme and the activity. So this enzyme is present both in the cytoplasm and the mitochondria. We can form these enzymes both in mitochondria and the cytoplasm. So this AST will be helping us to, that will be damage if it is increased more than 40. That is also reflects the damage to the hepatic cells. The hepatic cells is of liver. Okay, so this, if there is increase in the AST concentrations, so that is reflecting. If in the blood sample, if you see there is increase in the concentration of the AST, so that is reflecting it is damage to the hepatic cells. So But when compared to the ALT, that is alanine transaminase, so this AST is less specific to the compared to ALT, because this AST is present in distributed in all the cells like heart, liver, and majorly it is present in the heart and the liver and skeletal muscle. But when it comes to the ALT, that is alanine transaminase, this enzyme is majorly present only in the hepatic cells. This is very specific to the liver, specific only for the liver disorders. But AST is not specific for liver disorders. Okay. So AST that will be helping us in the diagnosis of various, along with the liver disorders. AST also helps in the diagnosis of the heart disorders, muscle, or even in the brain. So it's like a myocardial infarction during the heart attack. So we used to go for the AST test. So this will be helping us for the diagnosis of the myocardial infarction. So this is the cellular location of the enzymes. So this AST, that is cytoplasmic AST, as said, AST will be present both in mitochondria and cytoplasm. We can see the purple color. So and blue color, that is representing the presence of AST enzyme. Both it is present in the mitochondria and the cytoplasm. The red color was indicating the ALT enzyme, that is alanine transaminase. So this enzyme is only present in the cytoplasm, not in mitochondria. Okay. So when it comes to the GGT, that is GABA glutamate transaminase enzyme, this enzyme also comes under the liver function test. So this test will be usually enzyme is only present in the cytoplasm. This is the location of the cellular enzyme, that is liver enzymes. So when it comes to the alanine transaminase, that is ALT, is also known as SGPT. So that is this enzyme, the reference range for both SGOD and SGPT will be the similar, that is around the normal reference range for ALT is 0 to 40 units per liter. This is the normal reference range of SGPT. So when it comes to the distribution of this enzyme, it usually distributed in majorly in the liver and kidney and skeletal muscle and the arm. But the major activity of this enzyme can be found only in the liver and kidney. But when it comes to the minimal activity, so this SGPT activity can be minimal activity can be seen in skeletal muscle and the arm. This ALT test is specific for the liver disorder. Okay. But when it comes to the AST is helping us for the diagnosis of both liver disorder and even in the heart disorders like myocardial infarction. So this ALT will be helps in the diagnosis of various diagnosis, like it may be for the diagnosis of the myocardial infarction, cirrhosis of liver, hepatic cells. Majorly it helps for diagnosis of the cirrhosis of liver and the hepatic cell necrosis. This ALT will be diagnosis for helping for the diagnosis of these two disorders. So before reading the practical aspect, first you have to know what is this enzymes and where it is located and what's the function of these enzymes and how it is these enzymes will be helping us in the diagnosis of the disorders. So majorly these things will be learned in the practical aspects. Okay. So apart from that, so we have to know specimen collections. Okay. So this specimen collection is very important in the biochemical laboratory for the test analysis. Okay. So during during the clinical laboratories, we'll be having three different phases like the pre-analytical errors, analytical errors, and post-analytical errors. Okay. So if there is any errors during in the pre-analytical and analytical errors, so the test will be given a false positive result. For that, we have to be very careful during this analysis. Okay. So this specimen collections will be come under the pre-analytical errors. Okay. So to avoid this pre-analytical errors, we have to be very careful. The specimen collection will come under the pre-analytical error. So to avoid that errors, to give up exact correct results for the patients, we have to be very careful. So during the specimen collections, majorly for the estimation of serum, For the estimation of this ALT and AST concentration, we used to go for a specimen called serum. First, we have to collect the blood sample with the patients. After that, we have to centrifuge, and then we can collect the serum sample. By using this serum sample as a specimen, so we used to estimate the concentration of AST and the ALT. So always not only for the AST and ALT test, for the most of the tests we have to avoid the hemolysis. Okay, so this hemolysis should be avoided because it will be dramatically increases the concentration of AST. So if the sample is hemolyzed, so that will be increases the AST concentration. That may gives the false positive results. For that we have to avoid the hemolysis. So because if there is hemolysis of the sample, the red blood cells will be released for the serum sample. So that red blood cells will be contains the more 15%, 15 x of that percentage of this enzymes will be present in the RBC cells that may go for increase the concentration of AST in the serum sample. So that shows the false positive results. So for that we know that because AST is also present in the red blood cells. So to avoid, if it is hemolysis of the sample, the RBC will be released for the serum sample. So that will be the presence of AST in the RBC cells will be released to the serum samples. So that will be gives the false positive results. For that we have to avoid the hemolysis sample. Even in the post-acute myocardial infarction, so even in the post-acute myocardial infarction, we can see the AST concentrations will be levels will be we can see high. So rises even in the after post AMI around that will be rises after 6 to 8 hours, and that peaks will be reached at 24 hours. Okay, that will be returned back to the normal concentration by the 5 days. So after collection of the blood sample, so during the analysis of the sample, we have to know what is the history of the patient. That is importance of the importance will come under here. So always we have to know the patient's history. So whether the patient is in a post AMI condition or what. So for that we have to know. Otherwise we will give false positive results. For that we have to know the patient's history will comes here. So for that we have to know what is the condition of the patient. So in these conditions, after the post AMI, the concentrations will be rises after 6 to 8 hours. That will be reached maximum at 24 hours. So that concentration will be comes back to the normal by the 5 days. So for that we have to know all those things, pre-analytical errors. To avoid these pre-analytical errors, we have to know all those things. Apart from that, even in the conditions like acute hepatocellular disorders like viral hepatitis or cirrhosis, in these conditions the AST concentration highest concentration can be seen in these conditions. So this will be introduction about the EST and the ALT. In detail we will study the theory. So during the liver function test. So for the practical aspect, this is about the introduction. So now we will move on to the practical class. So now here, so we are doing the estimation of serum glutamate oxaloacetate transaminase, that is SGOD, by the IFCC method, that is KIT method. So the principle behind this experiment is same like as a transamination reaction. So here the SGOD that will be catalyzing the transfer of amino group, that is from the L-aspartate and from the alpha-ketoglutarate to form the oxaloacetate and the glutamate. You can see in the reaction. So there is a transfer of an amino group from the L-aspartate to the alpha-ketoglutarate in presence of an enzyme called serum glutamate transferase. So that will be forms the oxaloacetate and the glutamate. Further, the oxaloacetate will be reacts with the NADH molecule in presence of an enzyme called MDH, that is malate dehydrogenase. So that will be forms the NAD molecule. So this the rate of oxidation of NADH molecule to the NAD that will be measuring the concentration of SGOT activity. So the decrease in the absorbance will measures that will be representing the that is directly proportional to the concentration of SGOT in the given sample. Okay, whatever the oxidation of NAD plus has been formed in the reaction, so that will be decreases the absorbance. And that means the OD values will be directly proportional to the concentration of the SGOT activity in the given sample. Okay, this is the principle of this experiment. When it comes to the reagents, so the reagents which is required for this experiment is so this kit method, for the kit method we will always get the ready reagents from the company. For the clinical biochemistry, for the biochemistry laboratories, we will get the ready reagents. So in the form of reagent 1 and the reagent 2, that means R1 and R2 we will get in the ready form. Just we have to mix both R1 and R2. Okay, so that is the benefits from the kit method. So when it comes to the R1, so in the R1 that is in the reagent 1, we will be having L-aspartate and LDH, that is malate dehydrogenase enzyme, press buffer. So these are the three reagents will become under R1. In the R2 we will get alpha ketoglutarate and the NADH molecule. These are reagents which will be present in the R1 and R2. Okay. So to prepare the working reagent, so that will be the stock reagent. To prepare for the working reagent, so always we have to mix four parts of R1 reagent and one part of R2 reagent. For example, if you take 50 ml, you have to prepare for the 50 ml of this reagents. You have to take 40 ml of R1 reagent and 10 ml of the R2 reagents. Okay, for the preparation of the 50 ml. So based upon the requirement of the reagents, you have to mix both R1 and the R2, because once you prepare the working reagent, that will be stable only for 4 weeks. Okay, that is that will be stable for 4 weeks at 2 to 8 degrees Celsius. So for that, always based upon the requirement for that particular experiment, we have to prepare. So the specimen which is used for this kit method for estimation of AST that you are using here is serum. So when it comes to the test procedure, so the working reagent, first we have to take the 2 test tubes. So for that first, that is for the test tube. So for that we have to add 500 microliters of the working reagent, and the sample, that is serum sample, we have to add 50 microliters. So after that we have to mix well. So in the clinical laboratories we will be having the machines that will be aspirates the sample, and the absorbance will be read at 340 nanometers. So that will be gives the procedure. So when it comes to the wavelength, we are going for 340 nanometer or 670 nanometer. The incubation time temperature will be 37 degrees Celsius. There is no incubation time, and the mode of reactions will be the kinetic reaction. So the unit which is used for this enzyme will be unit per liter. This is the unit which is used for representation of the enzyme. So the slope of reaction, the absorbance will be decreased. Slope of reaction will be decreasing, and the reaction time will be takes around 60 seconds. Okay, so reference range will be 0 to 40 units per liter. So don't go for the factors and number of readings. This will be applicable in the clinical laboratory for the immersions. This is not much needed, just concentration in the reference range. So when it comes to the calculations, absorbance divided by the X factor which will be provided by the different of from the companies. That X factor will be applicable, will have to be applied there, and we will get some values based on that. We have to determine the results. Okay? So in the report we have to write the concentration of SGPT in the given serum sample, and whatever the values we are getting, you have to write it in that. Okay. So the clinical significance of this enzyme is, so this is the same like the transamination reaction I was explaining. So when it comes to the, we can see this enzyme will be elevated mainly in the EST. This EST levels will be elevated mainly that will be indicating the acute hemolytic anemia cirrhosis of liver, hepatitis. So in this condition, majorly AST levels can be elevated in 3 conditions. Apart from that, even in the hypotensive fraction and primary muscle disorder, recent surgery, acute pancreatitis or the inflammation of pancreas. So in these conditions, AST levels can be elevated. So clinical use of this AST is mainly in the evaluation that hepatocellular disorders, as I said. So this IST will be helping us for the evaluation of the hepatocellular disorders. In the acute myocardial infarction, that AST levels will be begins to rise within 6 to 8, and that will be reaches maximum at 24 hours. That will be returns within the normal within 5 days. So this AST will also evaluate the frequently seen in the pulmonary embolism and even in the skeletal muscle disorder like a muscular dystrophy, inflammatory conditions, even that will be causes the increase in AST levels. So even in the activities of serum alanine transaminase and aspartate transaminase are significantly elevated in several days before the onset of the jaundice and even in acute viral hepatitis. So even these conditions in the before onset of this jaundice and acute viral hepatitis, we can see AST and ALT activities with big concentration can be seen high. Even apart from that, so even in the ALT will be considered to be more specific for the liver disorder than the AST, because this AST it will be elevated in the cases of like cardiac, skeletal muscle injury, while in the ALT will not be increased. So when it comes to in the obstructive jaundice, so that will be increase occurs but usually does not exceed more than 200 to 300 international units per meter. So even in these, you can see when the hemolytic jaundice, the level of these enzymes can be normal. So this will be considered like during the clinical interpretations, we have to be very careful while interpreting the results. So when it comes to the estimation of serum glutamate pyruvate transaminase, that is SGPT by the kit method, the principle behind the experiment is seen as like that. So SGPT catalyzes the transfer of amino group between the L-alanine and alpha ketoglutarate that will be forming the pyruvate and glutamate in the end product. So here the pyruvate that will be formed will be going to react with the NADH molecule in process of an enzyme called lactate dehydrogenase, that is NADH, that will be formed in the oxidized NADH form. So the rate of oxidation that will be formed from NADH to NAD, that will be measures the activity, the concentration of SGPT activity in the serum sample. That will be directly proportional to the concentration of SGPT in the given serum sample. This is the principle. So here we can see the pyruvate, the amino group will be transferred in between the alanine and the alpha-ketoglutarate in presence of an enzyme lactate dehydrogenase that will be forming the pyruvate and the glutamate molecule. Again, that pyruvate molecule will be going to react with the NADPH molecule, so that will be formed oxidized lactate and the NADPH molecule. That NADPH will be representing oxidation rate of NADPH molecule will be representing the concentration of SGPT activity in the serum sample. So this is the principle behind the experiment. So when it comes to the reagents, so here for the reagent one, same as like that, we'll be having a reagent 1 and reagent 2. So reagent 1 will be consisting of tris buffer and L-alanine, lactate dehydrogenase. When it comes to the R2, we'll be having alpha ketoglutarate, NADH molecule. So this all the reagents will become under the reagents. So for the working reagents, same like that, for we have to add 4 parts of R1 reagents with 1 part of R2 as a like for the preparation of the, like if it is you have to prepare for 40 ml of the reagent, so you have to take 30 ml of this reagent and 10 ml of the part R2 reagent and you have to mix well. So that will be prepares the working reagent. So, like for example, I have given you an example. For 1 ml of working reagent, you have to mix 800 microliters of R1 and 200 microliters of R2. That's all that will be for the preparation of 1 ml of working reagent. So once we prepare the working reagent, that will be stable only for the 4 weeks. So based upon the requirement, we have to mix the reagent. So the specimen is seen here we are using serum sample for the estimation of the SGPT activity in the sample. So for the test procedure for this experiment will be, you have taken a test tube. For that you have to add working creation around 500 microliters, and the sample, that is serum sample, should be had around 50 microliter. So after that you have to mix well. So then we will calculate the OD by the absorbance. So that will be representing the concentration of SGPT activity in the given sample, and the OD will be taken at 340 nanometer. The test procedure here, incubation temperature will be the 70 degrees Celsius. Incubation time, there is no incubation time. Unit will be used as unit per liter. The wavelength will be 340 nanometers. So, and the reference range will be 0 to 40 units per liter. This will be reference range for the SGPT activity. So, and for the calculation, absorbance divided by the minute into factor, and that will reduce the concentration of SGPT. After that, whatever the values you are getting, had to write it in the report part, that is concentration of SGPT in a given serum sample. Based upon the values you are getting, have to interpret the results. So, this will be useless. So when it comes to the clinical significance, so this alanine transaminase or SGPT that will be helping us for the evaluation of hepatocellular disorders, like this high elevation of these enzymes will be majorly found in the extrahepatic, intrahepatic obstructive disorders. Even in the hepatocellular disorders, we can see elevated concentration of SGPT can be seen in these conditions. Even in the acute inflammatory conditions, like it may be of liver ELT, that elevations are higher that of AST, and that will be tend to remain elevated longer. Okay, apart from the during this acute inflammatory conditions, so the ALT elevations, the ALT concentrations will be higher than the AST concentration. So that will be representing that. So that will be give the indication of that will be causing from the acute inflammatory conditions. So even elevated levels of this ALT and will be indicates like alcoholic liver disorder, cancer of liver, hepatitis, or even in the inflammation of the liver, non-cancerous tumors of the liver, cirrhosis of the liver, death of liver tissue. So in these conditions, the elevation of this ALT can be seen. So this AST, the normal reference range will be 0 to 40 international per unit. So when during the for the this enzymes, this AST and ALT enzymes are the major markers for the hepatobiliary disorders. Okay. So when it comes to the AST, so this AST concentrations will be high in highest concentration can be seen in the viral and the toxic hepatitis. So the slight elevation can be seen in the obstructive disorders. So when it comes to ELT, in the viral and the toxic hepatitis, the highest concentration can be seen. But the thing is, in the obstructive disorders, the slight elevation will be there, but that will be more than the AST concentration. Okay. So in the obstructive, this ALT will be slight elevation, but when compared to the AST, slight increase concentration of AST is higher than but more than ALT will be higher than AST. So that will be helping us for the identification. This serum enzymes are major markers for the identification of the hepatobiliary disorder. So that will be helping us for during the interpretation of a disorder. So that will be completes the kit method. Now we will see estimation of AST and ALT by the REITMAN and FRANKEL method. So this method is for the this is a chemical method for the, but when compared to the kit method, the accuracy and precision will be low when compared to that kit method. Kit method will be having very high accuracy and precision. The results will be very accurate when compared to this. So here, because this will be comes under the manual method, so the manual methods the errors will be high compared to that. So the principle behind this enzyme here is the SGOT catalyzes the transfer of amino group that between the L-aspartate and L-glutarate, alpha-ketoglutarate, that will be forming the oxaloacetate and glutamate. So further the oxalate acetate that will be decarboxylases to form pyruvate and reacts with 2,4 dinitrophenyl hydrazine, that is TNPH molecule, that will be forming the hydrazone. So that hydrazone molecule, that is the end product, that will be forms the red color in the alkaline medium. So that will be directly proportional to the color, that the red color is directly proportional to the concentration of SGOT activity which is present in the serum sample, and the activity will be measured by using colorimetry at 520 nanometer. So for the ALT, the principle for this here is the ALT will be catalyzing for the transfer of an amino group between the alanine to the alpha-ketoglutarate, that will be forming the pyruvate and glutamate. Okay, so this pyruvate will be again going to react with the DNPH, that is 2,4 dinitrophenyl hydrazine, that will be forming the hydrazone. So that red color is directly proportional to the concentration of the SGPT activity in the given serum sample. So that will be measured by using colorimetry at 520 nanometer. So that is representing the SGPT activity in the given serum sample. So the reagent which is required for this experiment is phosphate buffer, substrate, pyruvate, standard color reagent, NOH, and the sample. The procedure here is the procedure here, we have to take 3 test tubes, test, control, standard. For the test you have to take so we have to take 4 test tubes, and for the buffers for all the 4 test tubes you have to add buffer solution around 1 ml. When it comes to the serum, you have to add 0.2 ml of serum sample to the test tube, and for the standard you have to add 0.2 ml. For the control and the blank you should not add any sample. And next you have to add distilled water around 0.2 ml for the blank. Then you have to keep it for the incubation at 37 degrees Celsius for the 60 seconds for 37 degrees Celsius for EST and 30 minutes for ALT. Okay, so the incubation time is different for EST and ALT. Okay, for 60 minutes for AST and 30 minutes for ALT. Okay, after incubation you have to add serum around 0.2 ml for the control group. Okay, so and after that you have to add reagent, that DNPH reagent, for all the 4 test tubes around 1 ml for each test tubes. After that you have to mix all the reagents and you have to incubate incubate for 20 minutes at the room temperature. After that you have to add around 10 ml of the reagent. 0.4 normal NOH will be added for all the 4 test tubes around 10 ml. So after that you have to take the readings at reading after 20 minutes. You have to take the readings at 505 nanometer. This is the procedure for the chemical method. The calculation part here is, so the concentration of AST by ALT is that is international unit, that is a unit, that is equals to the ODF test minus ODF control divided by ODF standard minus ODF blank, that will be multiplied with the 0.4 and 1000 divided by 0.2 into time in minutes. So this is the formula for calculation of AST and ALT, and after that you have to write the report as concentration of SGPT in the given serum sample, then you write the unit. Okay, next, concentration of SGOD in the given serum sample is equals to the, so next the diagnostic significance of the transformation. So levels of this usually, if it is more than 1,000 international units per liter, that will be, so this, if the concentration of AST and ALT, if it is more than 1,000 microliter, 1,000 international units per liter, so this levels can be occurs in the even in the acute viral hepatitis, toxin and drug induced hepatitis, or ischemic liver injury. So in these conditions, the concentrations will be more than 1,000 IU per liter. So when it comes to the most of the cases, like in the acute hepatocellular disorders like ELT, it will be higher or if that will be equal to the AST. Okay, in the hepatocellular disorders, ALT concentrations will be higher or that will be normal to the equal to the AST concentration. So this will be the diagnostic significance of the transaminases. So apart from that, even in the during the like for the distinction between the enzymes, these enzymes will be helping us in the for distinguish between the hepatocellular from the cholestatic jaundice. I said in the initially for the differentiation for to differentiate the cholestasis and for the hepatocellular, this transaminase enzymes will be help us for the distinguish between these two disorders. So when it comes to the hepatocellular jaundice, that will increase AST and ALT will be much more than 500 microliters. That will be representing the hepatocellular jaundice than in the cholestatic jaundice. When it comes to the cholestatic jaundice, this AST concentration will be more than 200 IU per liter. But when it comes to the if that comes to the hepatocellular jaundice, we can see AST and ALT concentrations will be more than 500 units per liter. So the persistence of elevated ALT and AST behind 6 months in case of hepatitis, so that will be indicating the development of the chronic hepatitis. So this all will be the diagnostic significance of this transaminase enzymes. So when it comes to the AST and ALT ratio, this normal AST ALT ratio should be around 0.8. If it is more than 2, that will be this more than, if it is more than 2, that will be this more than 2 concentration can be seen in the myocardial infarction, liver metastasis, erythromycin treatment, hepatitis, alcoholic hepatitis. So in these conditions, AST ALT ratio will be imposed at 2, but the reference normal will be around 0.8. So if it is low, if it is ratio is lower, that is ALT is higher, that means ALT concentration will be low. In that conditions, the AST ALT ratio, if it is less than 0.8. So in these conditions, we can see that is in the acute hepatocellular injury, toxic exposure, even in the cholestasis. In these conditions, the AST ALT ratio can be seen low. So I said there will be two forms of AST. Okay, one is mitochondrial AST and cytoplasmic AST. So when it comes to the mitochondrial AST, so that will be synthesized in the precursor form as a pre-M-AST, that is precursor mitochondrial AST. That will be converted into mature mitochondrial AST. So this mitochondrial AST and the total AST ratio, that will be a major marker for marker of chronic alcohol consumption. So during for the analysis of this chronic alcohol consumption, we will go for this mitochondrial AST and the total AST ratio. It will be act as a major marker for the identification of the chronic alcohol consumption. So this will be complete the practical. This is all will comes under the diagnostic significance of AST and ALT. So this will complete your practical class of estimation of AST ALT by KT method. Okay, thank you.